How was the material for you this week?
Oh, it was quite a lot of information for me, to be honest.
Huh?
But yeah, there's just so much to learn about all those neurons and all that stuff.
It was okay. I think I just have to rewatch the videos.
Okay, so you feel like you understood most of the points that were, because it's a little denser. This part is a little denser, I feel.
Yeah.
Yeah, it's definitely a little more complicated.
Okay. And of course, I had a great time making the questions for this stuff. You could tell, right?
Yep.
I'm sorry. All right, let me get a ruler.
You're not going to hit us with it, are you?
No.
No, we're not that lucky.
No, you're not that lucky. This ain't Catholic school yet. And now let's go to the material. Let's just do it. Let's see. Together with the endocrine system, the nervous system controls and coordinates what goes on within all our body cells. As we learned, endocrine glands secrete hormones in the bloodstream from where they find their way to target cells with specific receptors on them. Those then prompt the cells to act in specific ways. The nervous system works fundamentally in the same way, except that nerve cells don't secrete their substances, which now are called neurotransmitters, into the bloodstream, but rather send electrical signals along a cellular extension, which is like a cable, directly to the target cells, where a neurotransmitter, which are those chemicals, they are secreted and then picked up by specific receptors on the cells of where we want to have a specific action. The nervous system fundamentally consists of two parts: cables called nerves that send electrical impulses between the body and the brain the brain itself, which integrates all the information that is received. Can you name those two distinct parts?
Central, peripheral nervous system, Joanna.
Good. There you go. I know we get the quickie first here. So do you guys get that? Let me pull that up real quick. Oh, wait a minute. Yeah, there we go. I'm still confused how I have to deal with this stuff. Sorry about that. That's the slide. You got that?
Yeah.
Okay, good. So that's not, so if you guys, I want to make sure you pick up where the material was too hard or too confusing to understand, that we make sure we talk about those parts, okay? So don't let that just go sliding. Because the more you understand about what we talk about here, the easier the whole thing is later too. In order for the brain or the spinal cord for low-level decisions, that is, to control our cells functioning, it has to be informed to what is going on both internally as well as outside of our bodies. Some nerves, therefore, will carry informative messages into the brain, while others send command messages or commands to the body cells. We touched on that concept before discussing negative feedback loop. Remember? What impulses are carried in nerves carrying them to the brain, carrying the impulses to the brain?
Sensory.
Sensor. Good. Marcella, right?
Yes.
Oh, man. So that will be... So that will be this part here. The basic functioning is if you get poked, info goes in, that's sensory. We're going to process it. Info goes out. That's interpretation here, or integration. Info goes out, and that will be then motor impulses. So you want to distinguish there between sensory and motor. The endocrine system controls body functions that are ongoing and take time, such as growth or the cell's uptake of nutrients, for example. Sending hormones through the bloodstream is perfect for that. On the other hand, the nervous system controls body cell functions that require a quick response, such as moving the arm to turn a steering wheel in the car in order to avoid an accident. To send these signals with maximum speed, the body makes use of the concept of electricity, which is generated when negative charges, electrons here, travel through wires. What molecules in our body can generate electricity?
Ions, Marisol.
Ions, there you go. Again, that goes back, if you need to review that, review ionic bonds in chemistry, and that's where we talk about ions. So that's positive, negative charges, and in our body, the molecules that carry the negative charges are the ions, because they either have one too many or one too few electrons in their orbits. So again, any questions to that? Speak up. Number 4, I know people getting to start bullet holes that answer questions. That's great. Is this system making you guys do the homework more?
Yeah.
Good. We like that. I know some of you guys, if it's a little frustrating, let me know. If you don't get to talk, I can also pick people, but I think that would be gruffer. Okay, question 4. Magnets either have a positive or a negative side. The positive side is attracted to the negative on another magnet. 2 positive sides repel each other. Charges work in the same way. It allows to flow, if allowed to flow freely, would you expect positive charged molecules to move towards a positively charged field?
False,
Chris.
Alice?
False, Chris. If that's not clear, go to the fridge and take a magnet and turn it around and it doesn't stick. So then that's that positive and negative charge, basically. All right, and now there. Okay. A boundary separates charged fields. In the body, charges are carried on chemical molecules that have weight and mass. If they cannot move freely from one side to another, we can push them actively to one side and then use that established gradient to create a nerve impulse or electrical surges within the body. You remember that passive cell transport? If not, that's chapter 3 at the end. What cellular structure can establish such a boundary?
Plasma membrane.
Plasma membrane.
César.
Who? Okay, got it. Good. All right, let's look at the slide real quickly here. That's the plasma membrane. So again, remember, that's an oil film. These sticky things, those are oil chains, like from the fatty acids. And ions cannot move past that, but we can push all ions to one side or the other, and then we have such a concentration of a lot of them on one side compared to the other. And if we open a channel, like here, these are channels. If we open a channel, the area where there is many, many, many more, they all will flow into the other side, from one to the other side. And the sodiums will go in, the potassiums will go up, will go out, because when we try to establish it, when we push all those ions to one side or the other, all the sodiums we push to the outside of the cell, where we can. When I say all, I mean most. And all the potassium we push to the inside. So when a nerve impulse happens, there's so many more sodiums on the outside that we just need to open a gate and all of them flow inside very fast. It's like you don't have to, like if you think that of a real life example, it's like you have to take a bike up to a Merritt College. You have to pedal up. That's a lot of energy. It will take you much longer than if you go downhill, which you don't have to use your own ATP, your own energy. And so that's the same concept. And the potassium, what the body does, it puts all the concentration right on the inside of the cell. And so when we open those gates, all of them want to flow to the outside. And so those are real chemicals, real substances that carry a charge. That's where the sodium is plus and the potassium is also positive. And so we'll have you guys understand this so far, everybody?
Yeah, I think it's just I need to review I think in the first booklet we talked about like the cell and stuff. Just kind of review on that.
And which portion, the positive or negative charges, or the transport through the membrane?
I think the transport part is I'm just still kind of like rusty on it.
Yeah, okay. No, and that's good. We can review that real quick. Actually, I don't have that here, but what I want to make sure, and I think it's question 6 that goes with it. So what we have in the cell membrane is we have these specific pumps. And that is in booklet 3 as one of the transports. And so this specific pump is a sodium-potassium pump. And it pumps 3 sodiums every time it opens from the inside to the outside of the cell. And then it picks up 2 potassiums on the outside and brings them to the inside. And that pump just always works as a continuous. But that pump, it needs ATP. And so it's an active transport from that perspective. So it's kind of slow. But what happens is through that process, you keep pushing all the sodiums to the outside of the cell membrane and all the potassiums that you can to the inside. And then you end up having lots and lots of potassium on the inside of the cell comparatively to the outside. So the concentration is much greater on the inside. And the sodium is the opposite. They're all on the outside and not on the inside. And so when we want to make a nerve impulse, then we can go down here. Then we have these pumps. And so one pump is for sodium ions only, nothing else. And that when that opens, the sodiums all will rush into the cell because we have established that gradient, that high concentration right on the outside. So they all want to come in. And that fact that they have a charge on them, they have that positive on them, that means they with that ion, they carry a charge with them, and that will bring a little bit of an electrical change to the situation. Does that make a little bit more sense?
Yeah, it does.
Okay. Anybody else have? And some of people are texting because they can't get in. If somebody on your end can text them back that it's okay, that would be great. If you guys get that text. If not, it's okay. I just can't respond and talk at the same time. I get a little overwhelmed by that. Anywho, anybody else that has a problem when we talk about what we just talked about, these ions going to the inside and the outside? All right, good. Now let's go to the next question. Since the molecules cannot cross this boundary, the body can use ion pumps. So that's what I just talked about. So that to actively move specific ions to one side of the boundary, which is that cell membrane. This way, the body sets up a steep chemical gradient for specific ions. In nerves, sodiums are pumped to the outside of the cell and potassium to the inside. Sodium is the description on that slide over here. Sodium is Na and potassium is K. If that wasn't clear, that's one of those chemical things where they don't use the proper label for us. It's a German word. Anyway, so if you watch this, it might help. Did you feel that helpful? Did anybody watch this?
Yeah, it was helpful.
Okay, cool. Yeah, as you figured out, I could insert something. I learned a new technique on that, canvas at that moment. Anyway, when a nerve impulse is elicited, a specific channel in the plasma membrane open, allowing a specific type of ion to cross the membrane freely, which freely means passively, means we don't need ATP. Will those ions move towards or against its concentration field, up or down the concentration gradient?
Down.
Down. Who's that? Ebony.
Ebony.
Good. So that is kind of what I just described or tried to describe when we talked about these pumps and then when the channels open. Do you guys get that?
Yep.
Okay. So question number 6 is good for everybody?
Mm-hmm.
Okay. If I don't hear, then it's good. If one answers or so, please, that's great. If you do have a question, please speak up, even if it's a question later. Another concept is the one of voltage. It is the difference in charge between two fields in our body's discussion. That means the air are just inside and just outside of the cell/plasma membrane. If specific ion channels open and these little charged molecules cross the membrane, voltage changes. These detectable changes, that's electricity. the body uses specific amounts of changes in voltage as triggers to create nerve impulses that then are sent along nerves through the body. To me, that's pretty wild. To do this, the body's excitable cells need to have an established voltage while at rest. It's called a resting membrane potential. What structures in the body make sure that that RMP is kept up?
Sodium potassium pump?
Sodium potassium pump. Great. I got that, Alice. Thank you. And that is basically this thing here that we just kept been talking about for a little while. All right. These are important. These are fundamental concepts. So if you guys get these little questions and concepts, I try to have each question for one concept, then, you know, you're pretty good with the fundamentals here. Have you ever hit your funny bone, which is the inner elbow, and felt a shooting pain down the pinky? That pressure on what is called the ulnar nerve elicited it to fire. What was a nerve impulse, or also known as an action potential? What happens is that when a threshold is strong enough, sodium channels in the cell walls open to let a gazillions of these positive charged ions into the cell. That makes the inside of the cells charged more positive or less negative, you could also say. The voltage, so to speak, changes, that difference between the inside and the outside. And as a result, we feel a little shock when we hit that funny bone. This portion of the entire nerve impulse is called depolarization, named because the voltage or the polarity across the cell membrane is decreased. The other portion is called repolarization. Its goal is to send all those positive charges back to the outside of the cell in order to re-establish the original resting membrane potential. Which ion does the body use to cause repolarization?
K.
K. Hi, Kirsty.
Yeah.
Thank you.
It's K plus.
Yes, sir. Huh?
It's K plus.
Well, that don't count. That was good. Good. That is true, though. It is K plus. Did I write plus on there? So, number one, though, did you guys understand the concept of voltage?
No, but I will talk. But I wanted to talk after class about it.
Okay, cool. That's perfect. So that's the difference between the inside and the outside. So these negative and this positive just means the positive is a little bit out more on the outside and the negative is a little bit more on the inside. So when we open, when we want to shoot a nerve, when we want to depolarize, we want to make a nerve impulse, we open, or the body opens channels right in here, and all the sodium channels and all the sodiums want to rush in and bring positive with them. And that changes the relationship between this inside and the outside, and it becomes less big. That difference becomes less big, and that means it's depolarized. The D means less, right? And then when that nerve impulse is shut down, in order to tell the body to stop that nerve impulse, we're gonna open potassium gates, and then the chemical potassium, the ion, the physical structure will move outside because it's much more concentrated on the inside, and it carries a charge back to the outside. And so as a result, we basically have this polarized state again, and we have negative on the inside and more positive on the outside. So we're repolarized. Re means reestablishing that polarized state. Of course, the mess is that all the sodium is on the inside and all the potassium is on the outside. What are we going to do about that? What are we going to do about that? Any answer to that? Sodium potassium pumps. So that's where these puppies come in, and they work really hard and then reestablish that chemical gradient. And as soon as that chemical gradient is strong enough, we can fire the next impulse down the nerve. But that might take a few milliseconds. But, you know, of course, we don't care. That's like really fast, even if it's milliseconds. All right, that makes sense?
Yep.
Yeah.
It's some good stuff. Isn't that some good stuff? That's an action potential, what I just described to you. And so actually, let's go back to that action potential picture a little bit, because since we already did that. So that's where we talked about the RMP, the resting membrane potential. Oh, here's actually that pump where it shows you that 3 sodiums go to the outside, 2 potassiums come to the inside. And then here is that nerve impulse. And so I actually really like this picture here. It's probably the more confusing one, right? All right, let's go through this picture right here. You guys all understand this?
Kind of.
Good. That's a good start. So here is time. Down here is time. This is voltage. This here is how different the voltage is between the outside and the inside, this difference. And so what happens is here, when we see that red line, at some point, at this part of the cell, actually, which we'll talk about afterwards, that threshold is like you hit the funny bone, and at that point all the sodium gates open, and these are sodium gates. They open, that means open, and the blue is, and those rush in. They then bring all that positive energy in, or that positive ions in, and shoot up and bring the negativity to less than 0 in voltage specific terms. But it's depolarizing. It makes it less polar. So it brings the inside and the outside closer together when it is measured in voltage. And then at some point, here's positive 30, that's that point, millivolts. The sodium gates closed, the potassiums are closed, and at some point they open, and then all the potassium flows out, because we have such highly concentrated potassium right on the inside of the cell. And then then the voltage gets reestablished, and that's the repolarization. And then the sodium potassium channels are a little bit slow. No, the potassium channels are a little bit slow, so they undershoot a little bit, so they're hyperpolarized, they call it. But that is good, because that gives you an electrical moment where it's harder to make a second nerve impulse, and so the body can end up using that as a benefit. And then in the meantime, all up here, this shouldn't be right by repolarization. That's maybe not the right spot, but this is the sodium potassium pump, where sodium potassium pump restores the chemical environment. And that just is always floating and going. It's just when these channels are open, that's such a stronger force. It will neutralize those for a brief second, or for a brief millisecond. How is that in understanding?
Pretty good.
Pretty good. So you want to understand depolarization, repolarization, that they're linked to the channels of sodium and potassium, and that those are the ions that they both send a positive charge around. One brings it to the inside of the cell, and then one brings it to the outside of the cell. And so the one is involved in depolarizing, and the potassium then in repolarizing. So you need to understand that the chemical and the charge, they go together, but we're looking at the charge when we discuss voltage, and we're looking at the chemical, the ion itself, when we discuss which is more concentrated on which side of the cell membrane. And then you want to understand that sodium potassium pump is what establishes that concentration, that so much more sodium on the outside and so much more potassium on the inside. And what else do we understand about that? I think those are pretty much the 2 main points. If you get those, I think we're pretty good. Good. So with that, if I don't hear anybody speak up, I'll think we're moving forward, and you understand it. I'm very happy. If not, just speak up later. That's cool too. All right, that's crazy, I think. All of these nerve impulses or action potentials are equally strong. That means, for example, if we hear a sound, one impulse from the ear will communicate to the brain that we heard as sound, but it does not tell the brain anything about its intensity, such as loudness of the sound. How do neurons code for intensity of an impulse?
Frequency.
Melissa?
Frequency. Melissa. Frequency. Perfect. Everything else is a little less esoteric, I hope. So this is, oops. So we call that an all or none. The nerve impulse is all or none. It's not quite where I want it. That's all right. And so that means if you shoot a nerve impulse, that's always the same intensity and the same duration. And so you want to use multiple nerve impulses to say, oh, that was a really loud sound. And so you just shoot multiple signals, and then the body recognizes that as more intensity. And so you want to code with this frequency. Not too hard, right?
Nope.
Good. A nerve impulse is an electrical wave of opening and then closing sodium channels along the length of a nerve. It moves forward because when the voltage in one section changes sodium channels of the next segments open. Even though this process is fast, it can be sped up with a wonderful trick. Since ions are hydrophilic, they do not cross fat barriers. The body wraps phospholipid bilayer sheets around nerves. At those sections, ions cannot exchange, and the impulse has to jump to the next sections without any wrapping on it. What are those phospholipid bilayer sheets called?
Is it myelin sheets? Or?
Yes,
yeah?
It is myelin sheets, Nadiri. Perfect. And that we can understand right here with.
I have a question.
Mm-hmm.
Why is it spelled that way? Like
myelin?
No, like the sheets part. Why is it spelled like that?
Oh, I don't know. Do I spell it wrong? Hopefully no. No, I think it's spelled that way.
Yeah, that's how it's spelled.
It is like wrappings, like sheets of aluminum foil. So it's like sheets like that, but it's like these wrappings. Each of them goes around and around, and the more it goes around, the more it insulates it. And so in those sections, the ions cannot exchange because they only can penetrate that cell barrier with its channels in it, and that's one fat layer. But this is many fat layers, and so it cannot go through. So you will have much more action happening at those sections, which of course that's a good name too. Notes of Ranvier, where you don't have the myelin sheet, and so what happens is then the impulse will jump from section to section, which is called saltatory conduction down here. Salto, like jumping. Is salto, and that speeds up the nerve impulse and makes it much faster than if it just trickles down little by little. Good. 11. Look at that. We're moving right along. Now that we know the fundamentals of how a nerve's electrical impulse works, let's move to the anatomy. We first concern ourselves studying the small single cells within the nervous system, and then move on to study the brain and spinal cord. Two main cell types are the main worker bees in the system. Neurons are the ones that send electrical nerve impulses around the body, and that's the stuff we just learned, and they actually also receive stimuli or messages from other neurons and process the information received. The other set of cells help and support the neurons by giving them nutrients or defend against intruders. Or also, those are the insulating cells that we just saw that myelin sheath from. And then what are those helper type cells called?
Glia cells. Sorry, I know I already answered one.
Glia cells. I know you can hold. I know. I get it. I'm almost said open for everybody again. I think that's what we just did. Okay, round 2. Everybody can answer again once. Let me go find my little glial cell slide. Microanatomy. Why aren't you coming up, baby? Come on. Are you guys seeing the microanatomy slide?
Mm-hmm.
Neuron. Oh, okay, good. Then my computer is giving me a little grief. Now I got it. Okay, this is just the 2 types. One is the neuron itself, the electricity making thing. Cell, and then the glia cells. Glia means glue. That kind of, you can visualize it, kind of glues everything together and holds everything together and nurtures it to make sure everything's all right, as in nervous system. So that's good. Oh, look at that. We got one of those. Neurons come in many shapes and forms. Both our body Both our body's smallest and largest cell types are found amongst them. Generally speaking, we can describe 3 distinct areas of a neuron, match them to their function. All right, the soma. What's a soma?
In the metabolic center.
A metabolic center. All right, Ebony, you want to keep going?
Yeah.
Dendrite.
That one is in, they carry the impulse to nervous. And
carry it in, and then, I'm sorry. Then the other one?
Oh, sorry.
No, go for it.
Oh, that one's send nerve impulse.
There you go. Perfect. Good. Super answer. And I'm sorry if I cut somebody off. I don't mean to be rude. Sometimes I almost feel like I have to apologize to have to. Uh-oh. What did I just do? Wait a minute. Did I just do something? All right. There we go. That's the, yeah. So if I cut somebody off, I don't mean to be rude. But if I am, just speak up at the end. We can talk about it. Or so. Anyway, these are the 3 areas. So we got the dendrites. These are the branching-looking things that are all these little hair things, and then we got the soma. That's the body part on the inside. That's easy to be seen here in the schematic. And then soma means body. And then we got an axon, and that's the long tail-looking thing that goes all the way to the end where we have a little fizzling out stuff again. And that's the axon. That's actually, when we talk about the nerve impulse, that's happening in here. That's where we shoot that nerve impulse. That's like, if we want to move a muscle, we got one brain cell, that's one neuron that sits right underneath the brain here under the crust, and that's where the cell body is. And then we have a long axon that goes all the way to the spinal cord level of where the nerve comes out that goes to your muscle. So for the muscles in your toe, that's like almost by your butt. And that's where that nerve gets into the second nerve. So that axon is like a yard long, basically, of one nerve. And then there we have a second, then we have some dendrites, and then we have a second cell body, and then we have a second new axon that goes as a nerve all the way from the spinal cord level to where the muscle is that comes out of that spinal cord level. So for the foot muscles, that's pretty far down towards the butt. And so that's those are the 3 parts. I was always impressed how long those axons really are. That was always kind of like a wow moment for me for a long time. All right, in order to describe different types of neurons, we consider the extensions coming off the cell body. The most numerous type of neurons is mostly found in the brain and spinal cord. They have many extensions and are called multipolar neurons. Then we have some neurons with the cell body placed within one long extension. Those are called bipolar. Bi means 2, and we have 2 extensions coming out of the cell body. And lastly, we have cells that are sensory neurons, and the ones are carrying information. Those are the ones carrying information from the body through the spinal cord to the brain. And those neuron cell bodies are not protected within the CNS. In order to protect themselves, because cell bodies are very delicate alone, that's where metabolic stuff happens, and they have no protection at all. And so in order for them to protect each other, the cell bodies form many neuron groups together, protect the cluster of this vulnerable part of the cell. And it's strength in numbers question. And as a result of this, each of those neuron cell bodies only have one extension coming from it and going away from it and to it. What is that neuron cell type called?
Pseudo-unipolar.
Pseudo-unipolar. Mary Carmen. Hey, that's your first one, right?
Yes, it is.
Aha, perfect. Thank you. Good. So that long explanation, which I'm shooting myself in the foot having to read so long, my jaw always hurts afterwards. What am I doing? Sorry about that. There we go. Those are the 3 different main types. So when we actually describe a nerve, a general nerve, with dendrites, with a body, and with an axon, we describe the multipolar neuron. And those are the most numerous ones, and those are in the brain and spinal cord. And the bipolars are really only in the eye and in the nose. They're very rare. And the pseudo or the unipolar ones Pseudo-unipolar means because inside that little common off branch, you have both dendritic branches and an axon in it. And that's why they call it pseudo-unipolar. It's not really a proper term, I think, but that's the reason why. And those are your sensory or receiving neurons. So, if you need to match them up, that's where you want to do that. So that's just a piece of knowledge about this anatomy stuff. And then let's go to question 14. To a large extent, neural processing depends on counting up and interpreting all of the received stimuli. The chemicals released at the ends of the nerves are either excitatory or inhibitory to the receiving neuron. If all the excitatory messages combined are stronger than the inhibitory ones, the neuron will fire its own nerve impulse along that long cable called long cable called the axon. At its end, the impulse stimulates the release of its own chemical, which is that neurotransmitter. The neurotransmitter actually floats around in a narrow space to find its way to a corresponding receptor of the recipient of the message, which is the next neuron or the next gland. What do we call this area where the neurotransmission takes place?
Postsynaptic membrane.
Postsynaptic membrane, yes. And I think I was looking for that, or I think I simply also was looking for the word synapse. Understand, or is that question with that?
Is that the right answer?
You know what? Did you get it right or did you get it wrong?
No, I haven't seen. Well, it says that I had 3 wrong. I don't know which ones, but.
What question was that? It might be that I think the answer I was really looking for that was I think the main answer I was looking for was just the synapse, not even the specifics. So I have to see if I put all the specifics in. But I looked up on it. That's number 14, huh?
Yeah.
So it says, what do we call this area where the anti transmission takes place? So it says right here, the anti binds to corresponding receptors. So all that above that you mentioned is like, when it says bind, that means that's where like it gets done, right?
Yeah, no. I think I totally understand what you're saying. I just don't know if I, in my own head, when I wrote it, did that analysis to that level of detail, or if, you took it a level further than me.
I think I said the synaptic cleft because you said something in that description, you said something about the narrow space, and that's what it said for the synaptic cleft.
Okay, so those are all correct answers. So the question then is, was it confusing to you guys the way I wrote it, or were you pretty all right with that answer?
It was a little vague.
Was a little vague? All right, let's see. Yeah, see, of course, look at that. I just put synapse in. So I'm going to add on the synaptic cleft, postsynaptic membrane, review that question as a whole. Any other answers that you guys gave that that are different than synapse or postsynaptic membrane or synaptic cleft? No? All right, then let's just go B. Thank you for that. And sorry if I confuse you a little bit. But the concept is all right, right?
Yeah.
All right, good. That's the most important part. Neural connections between neurons can be formed and destroyed depending on their usage. Neural pathways connecting neurons in a sequence are most significant for nervous system function. The more a pathway is stimulated, the stronger it becomes. That is why I encourage retrieval exercises for learning this stuff. You know, that flashcard and labeling exercises where you have to reach and search information in your head, and that which means that always blah 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 blah. What am I saying? That when we have to reach for information in the head, then we hurt a little bit. It's not that easy, but when we say, "Oh, that's it," then we have fired that stimulus, and we have that. Why I said neurons that fire together, wire together. So strengthening of connections always kind of looking for an answer, and then knowing it, the more we do it, the easier it gets. And that strengthening of specific neural pathway happens when receiving neurons grow extensions back to the neuron that stimulated them in the first place. What do we call such a circuit? Anybody?
Reverberating.
There you go. Reverberating circuit. Where are we? Reverberating circuit. So this is to me, this is the most important part of this whole discussion tonight. Because if you understand that, then you understand all the stuff about neuroplasticity, all the stuff about when we are, the more we are anxious, the more we are depressed, the worse it gets, and we need to turn, if we want to turn those neural pathways around into the more positive, we are trying to be more open for experimentation and for looking at the nice sky on the, being happy, being grateful, actually. There's a really good TED Talk called Want to be happy, be grateful. And that's exactly this concept in terms of when we look at how neurologically this works. And so all the neuroplasticity talk, if you guys heard about that, it's a little popular at this point. This has all to do with these neurons that fire together, wire together. Meaning the more you fire a pathway, the stronger, the more wiry, the more real anatomical a pathway gets. Does that make sense?
Yes.
Because to me, that's good stuff. So that's why I put these in. I put this in with the pathway gets thicker. Good. I think that's enough talk about that. I'm sure I elaborated plenty everywhere else. A couple more concepts. When we look at the brain in particular, it is a very sensitive organ that needs to be well protected. It does that by being enclosed within the skull, which is a hard helmet-like bone. In addition, a thin layer of watery liquid surrounds the brain, protecting it further by absorbing shock and making it practically weightless. The problem with the brain being enclosed comes when it gets swollen. The skull provides a finite space that doesn't allow for much expansion. Infection is the biggest threat for that, besides shock trauma that the brain can get when we hit the head against something hard. Since infections travel most likely through the bloodstream, the brain's blood capillaries have processes from neural support cells that cling to them and seal them off so that the type of substances entering into the brain tissue is limited. What is that barrier called? I just said it. Anybody?
The blood-brain barrier.
Blood-brain barrier. There you go. So literally, you got these cells, these podocytes. Well, I think now they're actually astrocytes. They have these foot-like extensions in the kidneys. They'll be called podocytes when we talk about the kidney filter. That'll be interesting there. But they cling around these capillaries to seal them off, to make sure what goes in and out of the brains in terms of substances is very finitely filtered and controlled. So that's what that is about. Just an answer to why do we not get more infections in the brain kind of thing. When we look at nerves in our body, the ulnar nerve, for example, which gets excited when we hit our funny bone, most of them have cables in them that send sensory information from the body to the brain as well, ones that send motor impulses from the brain back to the body. What do we call a nerve that carries both sensory and motor impulses? Anybody?
Mixed nerves.
Mixed nerve, yes. So again, this feeds back to the discussion of negative feedback. Same concept. Information needing to go in, like you see something, most likely to the brain. We need to process it. Information going out. This just shows the spinal cord reflex, which we'll get to talk about at some point when we talk about the spinal cord. But the brain is the biggest processing center, and some very small stuff we can process in the spinal cord. But the important thing is here we have sensory nerves carrying information inward and motor nerves carrying information out. And we have some nerves that only carry sensory information, so we'll call those sensory nerves. We have some nerves that only carrying axons, that is, of of motor nerves. We call them motor, and then we have ones. Most of them carry both types of cables in them, and so we call them mixed nerves. And then the last item that we have here is the question of which part, if we get an injury, which part of the neuron can regenerate. Anybody?
Axon.
Axon. There you go. Microanatomy. Axon. If we, no, wait a minute. Where am I? We don't worry about that. Axon. If we have a break, if we cut some nerve, which there is mostly there's axons inside a nerve, is basically a whole bunch of these little cables, these little axons, and those can regenerate. But if you look at that, about 1 to 2 millimeters a day, which is like, I don't know what that is in inches, by the way. I should have looked it up. But it's like nothing. It's like very, very small amount of per day. But they can regenerate. And so if there is a damage to the cell body, I think we still consider that neuron will probably die out and cannot regenerate. All right, so that's it for that material.
I was still kind of confused, like with voltage, kind of, and like the gradients. Could you like re-explain the picture on page 7?
Okay, hold on.
It was the like the nerve impulse and like action potential and stuff.
Yes. Now we got it all out. Okay, page 7 of the booklet.
Yeah.
Oh shoot! Let me see where I have the booklet. Wait, I got it up here. Fundamentals. It's the fundamentals, right? It's in that part.
Yeah.
And we talked about voltage. Is the voltage?
Yeah, I was kind of confused with like the voltage, and then with like the nerve impulse, and then you were saying how it's like really important to understand it.
It is really important.
And I was like, I definitely do not understand.
Hold on. I am still stuck with where I am. Nerve impulse.
Oh wait, you just went past it.
Oh, good. Now you're seeing it. Okay,
right, one more down.
This?
Yeah. Oh no, where did it go?
This?
Yes, that.
All right, that's a cool shot. I'm proud of that picture. But which part is hard for you to speak? Talk through it and see how far you get in explaining a nerve impulse.
Yes, I don't understand any of it. When you're trying to explain it in class, I was trying to write it down, but then I was getting lost writing it down too. So I was like, I'll just come to you after class. And could you just kind of explain how you went through it in class so I could?
All right. No, that's good. That's good. So then let's see. So we have a couple of concepts here we need to understand. One of them is voltage.
Yes, please.
First, before voltage, I need to make sure you understand positives attract the negatives. Likes repel and opposites attract.
Yes.
You got that?
Yes, I understand that.
All right, cool. And then from there, we understand that we have these ions, these positive and negatives. And so that goes back with positive attracts and negative repels kind of thing. All right?
Yes.
And that goes where we understand then charge at the end. Because flowing, these positives and negatives flowing around create the electricity. Do you understand that?
Mm-hmm.
Good. So then it's a question of voltage. And voltage is the difference of positive and negative charges on the inside or between two fields. But here we talk about the inside and the outside of the cell membrane.
Yeah.
You understand that?
Yeah. Okay. I understood it more after class, but like this page.
This is hard, right?
Is where I got confused at.
Okay.
Yeah.
Okay. So the important is the electron, the charge, the ion part of the charge. You understand that?
Yeah.
Okay, so the charge is on the ion. Mm-hmm. And we compare the outside and the inside general charge right in those two spaces, not deeper inside, just right next to each other.
Okay.
And right on the, and so in a resting situation when the nerve doesn't fire, the outside has a little bit more positive charge to it, and the inside has negative, a little bit more negative as a whole when we measure it. This, in some ways, this does not have to do with potassium and sodiums. What it has to do then, where potassium and sodiums come in, is when we want to shift that difference in charge.
Mm-hmm.
So voltage is the difference in the charge. How many positives are here and how many negatives are here. Total. I mean, actually, no, it's even more than that. How many more positives versus negatives are right here, and how many more negatives versus positives are right here?
So that's like two
question.
Separate numbers?
Huh?
Is that, wait, are there two separate numbers for that?
Yeah, it's just a totality question. It's just like, is that area more positive or more negative?
Oh, okay.
Right? And so what we want to do in order to change, we want to change that difference.
Mm-hmm.
We want to bring some of those positives to the inside to
make it more
right in this area. To what?
To like make it more positive inside?
Yes, to just bring a few in to say, hey, something's happening. We make it more positive. We make a change in voltage, so to speak.
Okay, get that then.
Okay, cool. And then after that positive is on the inside, the other concept is that the positive is brought to the inside via a chemical. Sodium is a chemical.
Okay.
So then we're stuck. We got that physical chemical now on the inside. We have it more positive, but it's a physical chemical. It's stuck. We can't just take it back to the outside, right? That wouldn't work because we have that problem with that concentration.
Uh-huh.
When you think concentration, you think physical chemicals, like you think sugar cube in the water. When you just put sugar in the water, it sits on the bottom. You have to swirl it around because the concentration on the bottom is so much greater. And if you swirl it around, it wants to dilute, go through the whole water equally. That's what that means. The concentration wants to go from a higher concentration to a lower concentration automatically. The chemical wants to go from a higher concentration to a lower concentration automatically.
Mm-hmm.
All right. And so now when we have this change here, now what we got to do is to stop the nerve impulse, we got to carry that positive back to the outside. And we have a chemical. We have potassium that can do that. Potassium has a positive energy on it. It can bring it to the outside.
Okay, so it's kind of like trying to like rebalance it. Because I do understand like the repolarization. That's why.
Yes.
It's
so
like
that's rebalancing the electricity.
Okay.
When you say rebalancing the voltage, not rebalancing. When I think voltage, I think electricity. So rebalancing that voltage part of it, that positive negative discussion. What's not balanced after repolarize is the chemicals are messed up. There's potassiums on the outside more than on the inside, and so we couldn't have a second nerve impulse. Right? And so that's where that sodium-potassium pump comes in, which is here where constantly sodium is pumped from the inside to the outside of the cell and potassium from the outside to the inside. That's an ongoing process. And so when the nerve is not firing, we establish that situation, that environment. And that is both a chemical environment, meaning sodium and potassium chemicals, the ions, and a charge environment that's resting. And that's how much more positives are right on the outside versus on the inside as a total of all the chemicals.
Okay.
That's the resting membrane potential here.
Resting membrane.
All right. So far, so good?
Yeah. Get that so far.
All right. Now we basically, I think we're ready to go to this page now, the one in question. So this here is basically that voltage, that inside outside. So when it says, this should actually say negative here, 55, because that's the negative territory.
55.
So that means negative. That means the inside is 55 to 70 millivolts, which is just a little bit of electricity, more negative on the inside than the outside. That's what that means. And then when this shoots up, so that's where the red line is. The red line is that inside space, right inside the cell membrane. And then when it shoots up, meaning the more the positives are coming into the cell, so those sodiums are flowing into the cell, and that shoots up right on the inside. It shoots up that the inside becomes more positive in regards to the outside. So more positive chemicals come in. That makes sense, right?
Going up means that there's more sodium coming into this ion. Is it not the ion, but like the nerve?
More so, yeah, right next to, right, more sodiums come right in here, right, across the cell membrane, right at that part of that whole nerve, right?
Okay.
Yeah, this event describes every place around here. That's what that happens here. All right, so it's going to happen here, and then afterward it's going to happen here, and so forth until we reach the end point of a nerve. So, we understand. So this process here, changing the voltage, changing the inside to more positive an environment, means the sodiums come into the cell and taking with it the positive charge. And then at some point, and the point is positive 30 millivolts, you don't need to remember that. That's just at some point when the inside is more positive than the outside, the sodium channels will close and no more positive will come in.
Mm-hmm.
And we start crescenting here, and then the potassium channels open and they carry the positive back to the outside, and we go dive deeper again and become more polarized again. And basically reestablish the same environment we had before. We go down to more negative on the inside of the cell membrane than the outside.
Oh, okay. I understand too now. Like, so the 55 is the amount of negative that are inside. So the remaining is from the 70 is how many are outside, or how many positive are outside, left outside. Yeah,
right. You can actually, if you really want to just put a number here, you can, instead of voltage, you can just, it's a gazillion, but it's just a measure of a difference in the amount of numbers. When you say voltage, you could say the inside has 70 more negatively charged charges inside than the outside.
Yeah.
And so we take 70 in and we get more positive, and then that, you can just make it as a unit.
Yeah, now I understand. I was kind of confused with like the line and what, like, not what the numbers represented, but how we were like kind of looking at it. So now I understand.
No, that's good, because I also learned something, because actually you can, if you just put this is, instead of millivolts, this is like one more of these positive charged molecules on the inside than the outside, then you understand when you bring more positive ones in, it's going to change that number.
Yeah, that was easier for me to be able to look at it like that.
Right, it makes sense of the
if they're 55 negative inside, then either are positive.
And this is just a range, it depends on which nerve will fire in which range. Basically what happens, when this range is reached right here, when the nerve processes its own information, then it will shoot and fire that nerve impulse all the way down. And then these gates are all along that nerve. Well, actually, these gates, these sodium potassium gates, are most numerous where the nerve is not covered with one of those myelin sheets here, these bubbly things. And so that's how it can jump. Yeah, so next time you hit the funny bone, hopefully not too often, but you know what happens.